Have you ever wondered how scientists figure out which proteins are binding to DNA inside our cell? Let's dive into the world of ChIP, chromatin immunoprecipitation, with me in my YouTube channel Bioscope. Inside your cell, DNA doesn't float around freely. It's wrapped around proteins. Some of these proteins decide which genes should be active. So the question is, what is ChIP? ChIP is a powerful lab technique used to find out where exactly proteins like transcription factors or histones attach to DNA. This helps us understand how genes are turned on and off. Next, we can think that when the ChIP is used or where we can use the ChIP. Technically, ChIP is used to identify relative abundance of a specific protein or a specific protein modification at a certain region in the genome. ChIP can be used to answer a multitude of scientific questions involving the interaction of proteins and chromatin. For example, ChIP can be used to compare the presence of certain proteins at various loci, map the various proteins across the genome region of interest, or quantify protein binding to an inducible gene in response to the stimulus over time. Now, the next question comes, why ChIP is so important? ChIP is a super useful for studying cancer, gene regulation, and how our cell respond to stress or the signal. Now, the most important things come. How does ChIP work? So, the first step is cross-linking. Cross-linking reagents are used to fix proteins to the DNA that they bind. Formaldehyde is one of the most important reagents that are typically used to achieve this fixation. The second step is chromatin shearing or fragmentation. Chromatin fragmentation is essential for a successful ChIP experiment. Fragmentation of chromatin is necessary to solubilize the chromatin and allows for its co-precipitation. In addition, the resolution of the ChIP assay depends on the fragmentation of the chromatin, since DNA fragment size determines the resolution of the ChIP assay. This fragmentation can be done in two ways. One is enzymatic digestion that uses micrococcal nucleus, which cleaves DNA double strand between nucleosome to generate chromatin fragments, and second one is the sonication, where DNA fragmentation is done by sound waves. The step 3 is immunoprecipitation. Antibodies are used to capture a protein of interest and its bound DNA. In ChIP experiments, this resin is typically composed of ChIP grade magnetic saccharose or agarose beads conjugated to protein G. Antibodies bind protein A and protein G beads with various affinities depending on the species in which they were developed and the immunoglobulin G subtype of their heavy chains. Beads are typically incubated with antibody chromatin for 2 to 4 hours. Wash steps are needed to remove non-antibody bound chromatin, followed by reversal of cross-links and purification of DNA. The next step is the reverse cross-linking. Cross-links are reversed by high heat and high salt. Protein kinase K is also added to digest the associated chromatin proteins and added antibodies, allowing for more efficient downstream DNA purification. Next step is DNA purification and analysis. After the chromatin crosslinks are removed, DNA is either purified using classic phenol chloroform followed by ethanol precipitation method, or using column-based DNA purification kit. Once DNA is purified, several downstream analyses can be conducted, including ChIP PCR, ChIP qPCR, ChIP, and ChIP seq methods. ChIP PCR analyses are best for single gene analysis and can be used to amplify and quantify specific fragments of DNA in a rapid and cost-effective manner. ChIP analysis utilizes tiling DNA microarray chip to create a genome-wide high-resolution map of protein binding and protein modification. ChIP-seq analysis uses standard next-generation sequencing technology to align purified DNA with previously annotated whole genome to identify genome-wide protein and binding profiles. After this analysis, what is generated is called reads, which are strings of ATGCs. Then these reads are aligned with respect to a genome and peak is formed. Presence of peak means those regions of DNA are bound by proteins. So this is the whole process of ChIP. That's all for this video. If you found this video helpful, give a thumbs up and subscribe for more simple biology videos. If you have any question or suggestion, drop them in comments. Thank you for watching. Stay curious.